speakers and distinguished guests. Good morning to all of you. Morning, I'm going to talk a little bit about the assessment of risk associated with the consumption of food containing antimicrobial resistant bacteria. Can I have slides, please? Now, we all know we have been hearing about antimicrobial resistance, and um, it comes through various routes, as we have heard. It comes through water, it comes through food, and there is a lot of trade, international trade taking place in food. India is also a major exporter of several food commodities, particularly seafood. India exports seafood worth $7 billion US, and India is the largest supplier of shrimp to United States. And if we look at particularly fish and seafood, most of the production of these fish and seafood takes place in developing countries, and most of the consumption takes place in developed countries. And another thing in food that we need to keep in mind is today, food may be produced, the primary production may take place in one country, processing may take place in another country, and finally the consumption may take place in the third country. So if there is any antimicrobial resistant organism associated with food production and processing, so if there are any resistant organisms associated with food, it may be transmitted across borders. So there is a fear among food consumers that they may be getting antimicrobial resistant bacteria through imported food. Now, is it true? So let us look at how to make an assessment whether food can be a source of antimicrobial resistant bacteria. So one thing I want to also point out that when we are talking about bacteria associated with food and the transmittal of resistant organisms, we are not talking only about infection, but then what is the consequence of the presence of this antimicrobial resistance? So the consequence of an AMR infection could be that the infection goes on for a longer period of time, or there may be more frequent infections, or there may be treatment failure. So what are these resistant bacteria causing? So we need to make an assessment of this in food. Now in this slide, what I'm trying to highlight, particularly since I have been associated with the aquaculture and the aquatic food system, and aquatic food system is again another food product that has been often implicated in transmittal of resistant organisms. So people say aquaculture brings in so many resistant organisms. But if you see here, what we published in one of our FAO technical papers long ago, we try to highlight that all the resistant organisms, whether they have been selected in hospitals or in livestock sector or in agriculture, they all end up in the aquatic environment and finally ends up in the aquaculture system. So what we find associated with fish that is produced in a pond, if it is a resistant organism, it could come from any source. So we need to try to understand how resistance is coming. Often the farmers are blamed, saying that, oh, you are producing fish with so many resistant bacteria. But many of the entry of the resistant organisms may not be under the control of these production systems or the farmers. So we need to understand how the transmittal of resistant organisms take place and how to make an assessment of the risk associated with them. Of course, people already, several speakers have spoken about One Health, so I don't need to go into this. But then we need to try to see now, for example, in the fish, for example, in the aquatic environment, what are the types of resistant organisms that we can expect? So if we find resistant organisms, these resistance determinants may be naturally present. Now we all speak about the ESBL, these CTX, and what is the source of these ESBL? Now it is known that Now, in most of these human pathogens that are transmitted in the hospitals, probably this CTX gene is present in the plasmid. But it has been found in an aquatic organism, Cloivera, and it is chromosomal. So it is naturally present in certain aquatic organisms. And there may be also intrinsic resistance in several aquatic bacteria like Aromonas hydrophila, which has intrinsic resistance to ampicillin. Then, of course, we also find several enteric bacteria associated with food, even aquatic food like enterobacterials, which may be carrying resistant organisms that have been selected in other sectors. So there may be multi-drug resistant E. coli or Klebsiella. And there may also be resistant determinants in aquatic bacteria selected due to antimicrobial use in aquaculture. Previously, Indrani showed one slide where you could see the extent of use of antimicrobials in aquaculture. So if you see there, the proportion is only about 6% of use of antimicrobials occurs in aquaculture, and the largest portion I think occurs in the livestock sector and then human sector. So aquaculture also contributes to resistant bacteria, and then resistance determinants transferred, because all the time exchange of genes is taking place even in the aquatic environment. So when we find resistance in one organism, it may not be that it has been selected there. It could have acquired the gene during residence in the environment. So understanding the source of AMR in the aquatic environment requires a surveillance program. So you need a targeted surveillance to look at aquatic bacteria which are native to aquatic environment, and then targeted surveillance using bacteria that may be present in human or animal gut. So this will give us information on what is coming from, say, fecal contamination. And then targeted surveillance using environmental bacteria like Acinetobacter baumannii. So if we do a surveillance involving different types of bacteria which may be native to a particular environment or derived from other environments, we may get some information on how the transmittal is taking place. Now, regarding transmittal through food, Codex Alimentarius Commission is the international standard setting body for food safety. So they have come up with a guideline on how to do an integrated monitoring and surveillance is to be carried out in countries. And if you look at this recent paper in Lancet about the global burden, you can see the bacteria which are top in this list. You can see there are most of the bacteria may be derived from the environment. There are very few foodborne bacteria. Of course, E. coli can come through food, but most of the other Klebsiella also may come through food, but the other organisms are mostly derived from the environment. So food may be a source, but to what extent is it causing AMR infection? So that's what we need to look at. So this is the guideline from the Codex Alimentarius Commission, how to perform this risk assessment. So it requires a preliminary risk management steps which involves developing a risk profile and then ranking the risk profile to see which resistant organism we need to look at, and then see how the risk assessment is to be done, which is a scientific process, involves several steps. So what are the major steps involved in performing this risk assessment are outlined here. So now how do we identify whether AMR is actually a food safety risk? So this requires information which may be coming from surveillance data. So how many foodborne organisms are causing? Now we saw a paper in Lancet highlighting which are the ones causing high global burden. And then epidemiological reports, case reports, studies on interaction of microorganisms with environment through food production and consumption continuum, and studies on dissemination of AMR determinants in the environment, and science-based expert opinion. So to develop a risk profile, we need to develop a document which describes the AMR food safety issue, for which we have seen what could be the source of information, where we highlight what is the AMR hazard of concern and which is the antimicrobial agent, and what is the source of this AMR organism, transmittal route, pathogenicity, virulence, and characteristics of the resistance. What is the mechanism? Is it plasmid borne or is it present in the chromosome? or is it a result of cross-resistance, co-resistance? So all these aspects are to be highlighted in the risk profile. Then the information on the antimicrobial agent to which resistance is expressed. So class, whether it is used in humans or is used only in non-human use, extra label use, so all this kind of use information, related information. And about the food commodity, so how the food is being produced, what is the volume, what is the consumption, and what are the characteristics of the food, because that determines whether the organism will survive and will cause an infection in the consumer. And then what is the adverse effect that it can cause, so what are the characteristics of the disease, what is the frequency, severity, susceptible population, whether it is in only immunocompromised individuals or even healthy individuals may be susceptible, what are the epidemiological patterns, ethnic differences and the consequences of AMR on disease outcome, loss of treatment options, increased frequency, severity, prolonged duration and hospitalization and so on. So risk profile will also highlight the risk management options, because many times one may not be able to go to the actual risk assessment. The risk profile may be a sufficient document to take risk management action. So the risk management section could identify identification of the management options to reduce AMR hazard, and then measures to reduce the risk of selection and dissemination, minimizing contamination, cross-contamination, and evaluation of the available information and identify the knowledge gaps. So we need to always identify what are the knowledge gaps and what do we need to do further to improve the risk assessment. And once the risk profile has been developed, then make a ranking of this risk profile, then identify any particular combination of food and antimicrobial agent that needs further risk assessment, and commissioning this kind of risk assessment. So risk assessment has the steps of hazard identification, characterization, exposure assessment, and risk characterization. So this is same for microbiological food safety risk assessment, and even for AMR it is the same. So hazard identification step, we look at the microorganism and the resistance, and also at the antimicrobial agents. So it is similar to what we have looked at in a risk profile, but in a more detailed way, so that we need to try to see whether we can quantify the risk that is associated with consumption of a food and the antimicrobial agent associated with this. So hazard characterization again looks at the host and what are the adverse effects and what is the consequence, antimicrobial therapy and hospitalization, increased frequency of infection and treatment failures, or persistence of hazards in humans. So hazard characterization, because in the case of a microorganism and food, the numbers change because when food is processed, washed or cooked, there will be change in the microbial flora. So, and what are the characteristics of the food that influence the survival of an AMR organism in the food? And what is the dose-response relationship between the organism and the infection? So the mathematical relationship between exposure and probability of adverse outcome, that is the infection. So here the risk assessment may be qualitative, quantitative or semi-quantitative depending on the extent of data. So based on the information that we need or the data availability, so this may lead to increased frequency of infection or increased treatment failures or increased severity or prolonged duration of infection or hospitalization or increased mortality. So during hazard characterization, we need to look at these steps. And during exposure assessment, what do we see? So antimicrobial use selecting. So what is the reason why these organisms are present? Is it the antimicrobial use in a particular sector, or animal crop microbial factors affecting the dissemination of the microorganism, or other possible sources of AMR microorganism or resistant determinants in the target animal or crop? So that will tell us about the possibility of exposure. And then also, apart from the presence, we also need to consider the food related factors, that is food processing factors, and then consumer factors, how the consumers are handling food. Is it ready to eat food? Are they consuming it raw or after cooking? and the microbial factors affecting the transfer and maintenance of resistance. So that will lead to the information that is required for making the exposure assessment. So finally, what is the outcome? So if it is a qualitative assessment, we come with a result like there is a negligible, moderate or high risk. So this is just a qualitative assessment. And in risk characterization, we look more at the number of people falling ill, microorganisms attributable to the food source, duration of infection or death, and therapeutic alternatives, and comparison of public health burden before and after intervention. So if it is a quantitative, we need to think of some scoring. So if it is no additional risk, it could be assigned a value of 0, some additional risk value between 1 and 2, high additional risk 3 and 4, or very high additional risk 5 and 6. So this is some way of quantifying the risk. And then what are the management options? So implementation of good agriculture practice or good aquaculture practice and biosecurity, disease diagnosis, surveillance, restrict extra label use, and then also in terms of animal implement programs to minimize use of feed and ingredients. So waste treatment, farm waste management, post-harvest measures, reprocessing or recall of food that may be associated with these infections. So of course I won't go into this because there is no time, and I just want to highlight that our reference center is going to organize this workshop in November during the antibiotic awareness week. Thank you very much.
Thank you, doctor. Also time. I think what I understood as a pathologist and NABL assessor is risk assessment is the key. Thank you so much.